When infection causes pathologic changes that lead to disease, there are a number of associated signs and symptoms. A sign is an objective evidence of disease, as noted by an observer, like fever or septicemia or the chest sounds we might hear. A symptom, on the other hand, is subjective evidence of disease, as sensed by the patient. They might feel chills or fatigue or headache or nausea. So signs are much more precise than symptoms. For example, a brain infection may have a sign of bacteria that's in the spinal fluid, but the symptom could merely be a headache. Signs and symptoms of considerable importance in diagnosis are seen in the table to the right. We're covering many of these in our wiki posts. When a disease can be identified or defined by a certain complex of signs and symptoms, it's known as a syndrome. So let's begin by looking at the signs and symptoms of inflammation. The earliest symptoms of a disease result from activation of an inflammatory response. And the symptoms include things like fever, pain, soreness, swelling, perhaps edema. These are all non-specific defenses that are acting in our behavior. We may also see granulomas, which are solid masses of inflammatory tissue, and abscesses. Lymphadenitis, which is simply swelling of the lymph nodes, and on occasion we'll see lesions, which is the site of infection or disease, as in a boil or a break in the skin, perhaps a rash or other skin eruption. So, something to think about. If these are our non-specific defenses that are acting in our behavior that bring around these symptoms, why would we take over-the-counter remedies to remove those symptoms, to inhibit those symptoms? Could this really be a good thing to suppress our natural defenses? Something to consider when you're feeling lousy with some kind of infection. Should you let it run its normal course, or should you interfere with your own natural, non-specific defenses? How about some signs of infection in the blood? When we see changes in the number of circulating white blood cells, we consider these to be signs of possible infection. We can either see increased levels of white blood cells, leukocytosis, or decreased numbers in white blood cells, called leukopenia. Other signs of infection include the presence of a microbe or its products in the blood. When we have microorganisms multiplying in the blood and they're present in large numbers, we consider that condition septicemia. Microbes can be present in the blood but not necessarily multiplying, in which case they would be bacteremia or viremia. You might notice a certain root here. Of course, emia always denotes something to do with the blood. So septicemia means having large numbers of microbes multiplying in the blood. We also may see signs of the immune response, like antibodies that are in the serum. These are the basis of several of the serological tests. The root ser means blood also. On occasion, we may see infections that are asymptomatic or unnoticed. They're also known as subclinical infections. Now let's consider how pathogens exit the body. In most cases, the pathogen is released from the body through secretions, excretions, discharge, or sloughed off tissue. The release of the pathogen back out into the environment increases the likelihood that it can reach other hosts. So this is really the main mission for success of that particular pathogen. You have to keep in mind that that pathogen is trying to propagate itself and reproduce and increase its own population. Many times the pathogen uses the same portal to exit as it did as the portal of entry. Considering the exit portals is really important to epidemiologists because it influences the means of infection of a whole population. Pathogens may leave by respiratory and salivary portals through coughing and sneezing, talking or laughing. Skin scales as we shed billions of cells each day. They can exit in the feces. They could also exit through the urogenital tract and by removal of blood or bleeding. This is less common now as we seriously test blood before transfusions and such, but can still be transmitted through use of contaminated needles or open wounds. Now, once a pathogen has exited, the host may recover, but recovery does not necessarily mean that the microbe has been entirely removed. So the infectious agent could enter a dormant state called latency. That means that microbes, even though the host symptoms are removed, could still be persistent living inside those hosts. That microbe could periodically become active and produce recurrent disease. And even though the microbe's in a period of latency, it could still be shed and passed on to other individuals. Examples of persistent infectious agents include herpes simplex or herpes zoster, hepatitis B, AIDS, the Epstein-Barr virus that we've seen so much about in our wikis. Some diseases leave sequelae, which are long-term or permanent damages to tissues or organs. For example, meningitis can result in deafness, or Lyme disease might lead to arthritis, and polio leads to paralysis very often. So where do these pathogens persist? We call these areas reservoirs. This is the primary habitat in the natural world from which the pathogen originates. The source of a pathogen is the individual object from which the infection is actually acquired. The reservoirs could be living or they could be non-living reservoirs. First, let's consider the living reservoirs. A carrier is an individual who inconspicuously shelters a pathogen and spreads it to others without any notice. In the figure down here on the right, we can see some carrier situations. Asymptomatic carriers have no symptoms, however, they carry the disease. An example of this is with gonorrhea or genital warts. The individual who's carrying them may have absolutely no sign or virtually invisible signs. This figure also contains 3 situations of people who have had or will have the disease, but not at the time that they transmitted them. So you may have an individual that's in incubation. They don't even know they have the disease yet. You could have someone in convalescence who's recovering from the disease but still shedding pathogenic cells. Or in the case of a chronic carrier, chronic carriers have been carrying the disease for years and years and years, and it's in latency, but they don't express any symptoms of that disease. Each of these 3 have no symptoms at the times they transmit. Passive carriers have accidental transfer. That is, they don't necessarily pick up the disease themselves at all. This happens a lot in healthcare providers, where there's accidental transfer from the health provider to the patient. This is why providers need to be so careful to use aseptic techniques between care for each individual patient. Other living sources are animals as reservoirs. We've already seen animals like mosquitoes as vectors. Vectors are live animals that transmit an infectious agent from one host to another. The majority of vectors are arthropods, but as we've already learned, some larger animals can also be vectors. For example, mammals are vectors for rabies. There are biological vectors and mechanical vectors. Biological vectors actively participate in the pathogen's life cycle, whereas mechanical vectors transport the infectious agent without being infected. So a fly is a good example of a mechanical vector. It walks all over poop, picks up all kinds of bugs, and carries it mechanically to another source, maybe to your plate at a picnic. He never becomes infected or participates in the pathogen's life cycle. So even though he's a biological animal, he is not a biological vector. He's mechanical because he never becomes infected. Houseflies are the most noxious mechanical vectors. If you think about it, they feed on garbage and feces, they walk all over what they feed on, and then they land on your plate. Yuck, or they land on your melon. They carry over 20 bacterial, viral, protozoan, and worm infections. So really, absolutely disgusting. Zoonosis is when we see infections that are indigenous to animals, but have the possibility of being transmissible to humans. However, they're not usually transmitted to humans. In this table over on the right, you can see a number of the more common zoonotic infections. People in animal-oriented professions are at the greatest risk. We know of at least 150 zoonotic infections worldwide. They make up 70% of the emerging diseases worldwide. That means the new diseases that we're discovering. And mostly we're discovering them due to environmental destruction. So as areas become deforested or urban sprawl spreads into natural environments, animals have to find new habitat. They're forced into areas that are closer to humans, and thus their microbes can be passed much more easily to humans. Finally, let's consider non-living reservoirs in which pathogens can persist. As we've discovered, microorganisms have adapted to nearly every habitat in the biosphere, so they're literally everywhere. They thrive especially in the soil and water. Most of them are saprobic and benefit us by breaking down materials and recycling nutrients throughout the biosphere. However, human hosts that are in regular contact with these environmental sources may pick up some of the not so good, not so favorable pathogens. There are a few, but they're still easy to pick up. Before we move on, let's have a quick concept check. Which of the following is a symptom? Let's consider, again, what is a symptom? A symptom is something that a patient feels, whereas a sign is something that is measured much more objectively. So let's look through this list. Elevated white blood cell count, fever, nausea, and swollen lymph nodes. So white blood cells we can count, so that's a sign. Fever, we can measure that with a thermometer. Nausea, how sick does someone feel? This one might be good for us. Swollen lymph nodes, we can also feel from an outside observer, so it's a sign. So nausea is really the only viable sign in this one. Now let's move on to think about how we acquire and transmit infectious agents. When an infected host can transmit the infected agent to another host and establish an infection in that host, the disease is considered to be communicable. Keep in mind, it's the microbe that's communicated, not the disease. So there's a little bit of technicality in terminology here. The transmission can be direct or indirect, and we'll spend some time exploring those different mechanisms. Now, if the agent is highly communicable, it's considered to be a contagious disease. Things like measles and influenza are contagious. Now, leprosy, on the other hand, is only weakly communicable, so we don't consider it contagious, although it can spread through a population. It's communicable diseases that will be the focus of this section. However, before we move on to that, let's take a brief look at what non-communicable diseases are. These are diseases that do not arise through transmission of an infectious agent from host to host. So they're acquired through some other special circumstance, perhaps like a puncture wound by a nail that's infected with tetanus. We're not going to pass that tetanus onto another host. It's just an individual isolated case. Sometimes we'll see a compromised person invaded by his or her own microbiota. Again, this is not going to be passed from host to host. If an individual has accidental contact with a microbe in a non-living reservoir, again, like tetanus, it's a non-communicable. So that should clarify some of our terminology. Now let's look at transmission. There are many patterns of disease transmission. They can be direct or indirect. They can come from animate or inanimate objects. We can have horizontal or vertical transmission, where horizontal is from one host to another host, and vertical is from a parent to an offspring, either through the ovum, the sperm, the placenta, or milk. We're going to divide the microorganisms into two major groups. One, direct transmission, where there's some form of direct contact, and the other group, indirect, which involves a vehicle. So the transmission moves from one host to the other using some sort of intermediate vehicle. In direct, we have to have some kind of direct contact between the skin and mucous membranes of an infected person with the new infectee. So there is no intermediate object. So direct or contact transmission comes through touching or kissing or sexual intercourse. It could be a bite by a biological vector because we're obtaining the pathogen directly from a vector. Vertical transmission, again, prenatal can come across the placenta, or perinatal shortly after birth in the form of breast milk. Also, direct transmission might not seem so obvious is droplet contact. Droplet contact involves picking up particles from someone sneezing or coughing. The key here is that the person or the people involved are in each other's presence. So even though the droplets are passing through the air, they're passing directly to the new infectee. When we consider indirect transmission, we must have an intermediate conveyor, a vehicle where contamination of inanimate objects or food or air carries the pathogen from one host to another. So a vehicle is any inanimate material commonly used by humans that can transmit infectious agents. So food, water, biological products, or formities. Formities are contaminated objects like doorknobs or telephone or remote controls or faucets. The list goes on and on and on. But basically, a formite, the singular form, is any inanimate object that harbors and transmits pathogens. Again, the list is huge. I'm sure you can come up with hundreds all on your own. We can also get indirect transmission from food poisoning, either pathogens found in the soil or from a handler of the food, or from a mechanical vector, again, like the flies come and land on your melons. Also, we could have indirect transmission through an oral fecal route. This is very common. We see it on contaminated toys or food handlers with inadequate personal hygiene. We see a lot of oral fecal route transmission occurring on multiple day river trips when people are not as rigorous about their hand cleaning routine. And I'll tell you, this stuff passes fast. All it takes is one person to not wash their hands between bathroom visits and get their hands on your food. Everyone on your whole trip can get sick, and that really ruins it for people, especially when you're away for, say, like 3 weeks on the Grand Canyon. Anyway, air can also be a vehicle for indirect transmission. Now consider that outside air cannot provide the nutritional support for microbial growth, but indoor air can, especially when it's in enclosed spaces. Droplet nuclei are different than droplet contact. Droplet nuclei are transmitted some distance by air. They are dried microscopic residues of sputum or mucus, and they remain suspended in the air in the room. They can be passed to a new host, either geographically distanced from each other or chronologically distanced. So let's say one person comes into the room, sneezes, all of that material dries and remains suspended. And 2 hours later, another person comes into the room. We're now chronologically distanced, but that individual could still inhale these microscopic particles. So droplet nuclei transmission is an indirect means from sneezing or coughing or even speech. But droplet contact is where both individuals are actually present in the same time and space. Aerosols are another means of transmission. These are fine dust or moisture particles that actually contain live pathogens, and again, allow transmission through geographical or chronological distancing.